Hello everyone, this is Minoy Cristobal, a doctorate student in Dr. Hans Stein Monogastric Nutrition Laboratory. Today I will be discussing one of our research titled Effects of Diet Protein Concentration on Apparent Ileal Digestibility of Starch and Standardized Ileal Digestibility of Protein and Amino Acids in Diets Fed to Growing Pigs. Because of the demand of a more precise nutrition, synthetic amino acids started to be used in animal diets. The industrialization of some key amino acids led to the development of the amino acid industry. For instance, the first amino acid that was industrially produced was methionine in the 1950s, followed by the commercial production of lysine in the 60s. A few decades later, the industrial production of threonine and tryptophan started, and after 2015, all indispensable amino acids are available in a synthetic feed-grade form. This availability allowed nutritionists to use synthetic amino acids in pig diets and therefore reduce the inclusion of protein ingredients such as soybean meal and have diets with reduced crude protein. Research has been conducted to determine the impact of low protein diets, and in general, authors observed a lower nitrogen excretion, equal growth performance as in diets with no synthetic amino acids, and it has been suggested that reducing crude protein in diets increases net energy of diet because less energy is required for the amination of excess amino acids. However, this is just an assumption, and studies that actually determine net energy in low protein diets are limited. In a previous research conducted by our group, we formulated corn soybean meal diets with reduced crude protein from 17 to 10%, supplemented with synthetic amino acids, and we determined net energy in these diets through indirect calorimetry using group housed growing pigs. It was expected that the net energy in these diets increased not only because less deamination may occur in the low protein diets, but also because, according to the NRC tables, corn contains more net energy than soybean meal, and we had more corn in the low protein diets. However, in the experiment, we observed that the net energy in diets did not seem to increase or decrease. So this would indicate that there is something that is different from NRC values. Then the question is, why is that we cannot pick up these differences? Is that because there are differences in digestibility of a starch or differences in digestibility of amino acids among these diets? And research about this in low protein diets is limited. Therefore, the objective of this research was to test the null hypothesis that reducing diet crude protein by reducing soybean meal inclusion and increasing corn inclusion and synthetic amino acids does not affect the apparent ileal digestibility of a starch and does not affect the standardized ileal digestibility of protein or amino acids in diets for growing pigs. Moving on to materials and methods, we used the same 6 diets as in our net energy experiment, but we had to add chromic oxide to determine ileal digestibility using the index method. Now, for diet 1, it contained corn and soybean meal and no synthetic amino acids, and this diet had a crude protein of 17%. In diet 2, we added 3 synthetic amino acids, lysine, methionine, and threonine, and with that we reduced soybean meal inclusion so that crude protein in the diet was reduced to 14%. In diet 3, we added 4 synthetic amino acids, lysine, methionine, threonine, and tryptophan, and reduced soybean meal inclusion to have a crude protein of 13%. We followed the same logic for the next diets where we added more synthetic amino acids, and as we reduced soybean meal inclusion, we also increased corn inclusion so that we reduced crude protein in the diets from 17 to 10%. And these diets met the requirements for standardized ileal digestible indispensable amino acids and vitamins and minerals for growing pigs. And we also used a 7 diet that was a nitrogen-free diet to determine basal endogenous losses of amino acids. Now let's take a quick look at the analyzed composition of diets, except for the nitrogen-free diet. Values are represented by different colors for each diet from left to right, from the 17% to the 10% crude protein. We observed that the analyzed crude protein was close to the formulated crude protein. And analyzed starch in diets increased as we reduced crude protein, and that was because we increased corn inclusion in diets. For the analyzed concentration of lysine, threonine, tryptophan, and valine in diets, in general, we observed that the concentration decreased as we reduced crude protein in diets. This was because we added more synthetic amino acids in diets, and these are 100% absorbed compared with the amino acids from soybean meal that have a digestibility of around 90%. Therefore, a slightly less concentration of these amino acids were needed to meet the requirements for amino acids. However, concentration of methionine increased, and this was because as we reduced soybean meal inclusion, we also reduced the cysteine concentration. Therefore, we had to increase synthetic methionine to furnish for methionine and also for cysteine. Moving on to animals and experiment design, we used 7 barrels of around 38 kilos that had a T cannula installed in the distal part of the ileum. We allotted them in a 7x7 Latin square design with 7 diets and 7 periods for a total of 7 replicate pigs per diet. Pigs were fed 3 times their ME for maintenance, and each period consisted of 5 days of adaptation to the diet, followed by 2 days of collection of ileal digesta samples for 9 hours each day. As for our stat analysis, we used the mixed procedure in SAS, where diet was the fixed effect and period and pig were random effects. We used contrast coefficients to determine linear and quadratic effects of reducing crude protein from 17 to 10%. Looking at the results, the apparent ileal digestibility of a starch was around 95%, and we observed no effects of reducing crude protein in diets from 17 to 10%. For the standardized ileal digestibility of amino acids, for lysine, the values ranged from 80 to 90%, and we observed a linear increase in SID of lysine as we reduced crude protein in diets. Similarly, for methionine, the values were around 90%, and we observed a linear increase as we reduced crude protein in diets from 17 to 10%. Similar linear increase was observed for almost all other indispensable amino acids. When we determine the SID of total indispensable amino acids, we observe again a linear increase as we reduced crude protein in diets from 17 to 10%. And the same linear increase was observed for dispensable amino acids and overall standardized ileal digestibility of crude protein of diet. Now with these results, let's revisit our hypothesis that was that reducing diet crude protein by reducing soybean meal inclusion, increasing corn inclusion, and synthetic amino acids does not affect apparent ileal digestibility of starch, and we cannot reject this null hypothesis, one, because we observe no effects on AID of starch. However, we reject the null hypothesis 2, that was that no effects on SID of crude protein and amino acids, as we clearly observe an increasing effect on SID of protein and amino acids as we reduce crude protein in diets for growing pigs. These results, especially the increase in SID of crude protein and amino acids, explains why nitrogen excretion has been demonstrated to be reduced in pigs fed low protein diets compared with high protein diets. But these results do not fully explain the lack of effect in net energy in low protein diets observed in our previous experiment. But combining these results, we could say that the lack of impact observed on net energy in low protein diets is not because differences in starch digestibility and definitely is not caused by a reduction in amino acid digestibility. So, what can it be then? For one hand, it could be that the net energy in soybean meal is equal or closer to the net energy in corn, and this is also in agreement with other recent research that has demonstrated that there is no difference in net energy between corn and soybean meal. And for the other hand, it could be possible that besides the high absorption of synthetic amino acids, close to 100%, as we indirectly demonstrated it with our experiment, but also synthetic amino acids might have a more rapid absorption compared with amino acids from intact protein. And protein synthesis only occurs if all the indispensable amino acids are present in the cell at the same time. Therefore, if synthetic amino acids are absorbed more rapidly, and because cells cannot store free amino acids, then they might be deaminated before all other amino acids reach the cell, leading to a reduced protein synthesis, which might contribute to have less net energy in the diet. If this hypothesis is correct, it implies that there might be a limit in a quantity of synthetic amino acids that can be used in pig diets. With that, I would like to acknowledge the United Soybean Board for their support on this experiment, and also thanks to all Dr. Hansenstein Monogastric Nutrition Laboratory members. And if you would like to know more about our research, we invite you to visit our website. Thank you.